Okay, yeah, I'd like to thank the committee for this invitation and an opportunity to present. Okay, so here are my disclosures. So effective combination immunotherapy is really required for relapsed refractory patients, but also to deepen therapy-induced responses for CLL patients. And unexpectedly, no activity was recently seen in an anti-PD-1 checkpoint therapy trial for relapsed disease. And many patients also exhibit very low responses to the CAR T-cell immunotherapy approach. So this clinical evidence so far is indicating that there are multiple immunosuppressive mechanisms operative within the environment that are contributing both to the immune deficiency of CLL, but also a barrier to these novel therapies. So my group are interested in understanding the different obstacles that cytotoxic T cells, dendritic cells face in the CLL microenvironment. And the long-term aim is to use this knowledge to then develop novel therapies to overcome these obstacles. So firstly, for the first part of my talk, I'll just describe quite briefly unpublished data, which is now ready for submission. And here we've been using modeling to look at the ability, firstly, of checkpoint inhibitor drugs to interfere with T-cell tolerance. So this first data slide shows that when we treated CLL patient samples with anti-PD-1 immunotherapy drug alone, what we observed was a small but non-significant increase in T-cell killing function against the patient tumor cells. This led on to us to investigate further the expression profile on the patient T cells, and interestingly, we found that PD-L1 expression was also expressed on a proportion of patient T cells, particularly following activation. Now, when we went on to perform cytotoxicity assays, we see that anti-PD-L1 actually interestingly performs better than anti-PD-1 monotherapy. But notably, we observed that both checkpoint inhibitors alone only partially reactivate or activate the patient T cells, and we know this because we're always comparing to age-matched healthy donor T cells. So this led on to Confocal Studies, and here we're looking at patient lymph node biopsies, and here we're looking at the checkpoint network and the immune landscape. And our initial observation was that the CD8 infiltration is heterogeneous, but generally low in these CLL lymph nodes. And interestingly, we found that the PD-L1 expression was largely associated with the T cells, which is shown in this diagram here, the T cell with PD-L1. And this really contrasts a lot to the tumor cells, which exhibited very weak expression of PD-L1, and of course contrasts with the situation in Hodgkin's lymphoma, where we know that the tumor cells express high levels of PD-L1. So this really provides evidence that a low infiltration of profoundly exhausted CD8s, as well as a low inflammatory microenvironmental state, appear to be the challenges in CLL, and this argues for the identification of additional immunostimulatory therapy. So we've been using avadamide here. This is a novel cereblon modulating drug, and this data slide just illustrates that, again, using modeling, that this drug activates the T cell immune synapse. You can see in red repair of F-actin synapses coming through. But interestingly, we also saw a concomitant increase in PD-L1 expression shown in green following treatment with avadamide. So this then led on to cytotoxicity models where we've paired avadamide with either anti-PD-1 or anti-PD-L1 combination therapy. And this combination effectively reactivated the patient T cells and maximized tumor kill in our assays across different patients. So next, because we're realizing the importance of the T cell compartment, we performed RNA sequencing. This is highly purified patient T cells treated with these immunotherapy drugs. And what our gene expression analysis revealed is that the top functional gene categories were linked to type 1 and type 2 interferon signaling responses in the patient T cells. And this was present both with avadamide, but also in the combination immunotherapy groups, and independent from PD-1 or PD-L1 alone. We also detected genes enriched for T cell proliferation, chemokine signaling, including CXCL10, and which is linked to the interferon response, as well as cytoskeletal and cytotoxicity repaired pathways. We then went back to a differential gene expression data and showed that the interferon type 1, type 2 pathways, as well as the chemokine pathways, are deregulated in patient T cells compared to healthy donor T cells. And overall, what our avadamide data is showing is that these signatures, the drug signature, is normalizing these T cell pathways. So lastly, we've used the patient-derived xenograft model that Nick described. Rather than pre-activating T cells, we've introduced PBMCs into busulfan-primed NSG mice. And this allows establishment of disease. And I'll just highlight very briefly here that in vehicle, the CD8 cells are present in low numbers and excluded from the proliferating tumor area. So we've treated these mice with avadamide and anti-PD-L1, which I'll highlight here. And hopefully in green here, you can see that this combination was enhancing the recruitment of these killer cells into the established tumor cells. So we were treating established tumors. In harmony with RNA-seq, we see that PD-L1 goes up in the microenvironment of these mice, including particularly T cells as well as tumor. And I'll just draw attention to CXCL10. We saw a dramatic upregulation of this on the CD8 T cell compartment with avadamide therapy. So treating these mice with avadamide and anti-PD-L1 maximally reduced the tumor volumes with therapeutic treatment. Okay, so hopefully this will work. So just to summarize this first part of my talk, what I'm illustrating here is that interfering modulating drugs like avadamide can potentially drive a proliferation response in the T cells. This can induce CXCL10 expression in the microenvironment, which can then recruit the CD8 cells into the tumor area. This is part of a feed-forward cascade. It complements with anti-PD-1 or anti-PD-L1 antibody, and this combination therapy is activating the patient T cells to maximally kill tumor cells. Okay. So really in this current immuno-oncology era, there's certainly potential to harness exhausted patient T cell function. The second part of my talk is really describing what we believe is additional barriers to therapy, and this is additional immunosuppressive mechanisms within the microenvironment. And this unpublished work, which is progressing in the group, is studying or modeling the fibroblast microenvironment in CLL as well as other lymphomas. So to do this, we're looking beyond the classical tumor cell immune model and asking the question, what lies beneath? So we've been studying immunologically specialized lymph node resident myofibroblasts, which are called fibroblastic reticular cells, or FRCs. And FRCs are known to produce and remodel the extracellular matrix to essentially create a reticular highway, which I've illustrated in this cartoon, which is used by T cells and dendritic cells to interact and, of course, form adaptive immune reactions. FRCs have a role in regulating both adaptive and innate immune cells. Importantly, what occurs in the chronic cancer scenario has been understudied in these lymph node fibroblasts. So here we're using modeling to ask the question, what happens when tumor cells are exposed to fibroblasts? What's happening, what's being modulated in these myofibroblasts? So we're using established 2D, 3D culture models where either human or murine CLL tumor cells are co-mixed with previously healthy fibroblasts. So following the co-culture, we then perform phenotypic and functional assays. And just very briefly here, what we see in health is that the FRCs display this typical highly contractile phenotype. But interestingly, when we expose the same FRCs to the tumor cells, they take on this activated phenotype, and this causes the fibroblasts to take on this stretched morphology. Now, interestingly, we've managed to identify primary CLFRCs from an excess lymph node biopsy. And here, using 3D models, we're illustrating that these primary CLFRCs also exhibit this stretched, elongated phenotype in the 3D model. Perhaps not surprisingly, these fibroblasts are promoting tumor survival and keeping the tumor cells alive in culture with this data here. And fascinatingly, what we see with longer-term culture is the ability of the tumor cells, CLL tumor cells, to actually downregulate podoplanin, which is a transmembrane glycoprotein expressed on fibroblasts. And this downregulation has important consequences for the immunomodulatory potential of these fibroblasts. So importantly, with all our modeling work, we always verify our data in vivo. Here, we're looking at the healthy FRC network, which is stained with podoplanin in white, alpha SMA in red. And this is an organized, restricted, healthy FRC network in non-malignant lymph nodes. When we look at our patient lymph node biopsies, we actually see that the network is significantly remodeled, particularly with the alpha SMA stain. And quite dramatically, we see a loss of podoplanin across the reticular network. We've also confirmed with preliminary studies that the TCL1 model recapitulates this alpha SMA remodeling. So we're interested in the lab to identify the tumor factors that may be activating these fibroblasts and altering their immunomodulatory phenotype. And I'll just draw attention briefly to a few. We've seen surface CLEC2 on tumor cells. We also believe intracellular TNF-alpha, IL-4, and IL-13 are playing a role within the tumor B cells in modulating these fibroblasts. So we use RNA-scope, and here's this TNF is being associated with tumor cells within patient lymph nodes. And this is preliminary data showing that if we block IL-4 receptor on the fibroblasts, we can partially prevent tumor cells or recombinant IL-4 from downregulating podoplanin on the fibroblasts. So next, we've been interested in examining what is the transcriptional consequence for these altered fibroblasts. So here, transwell co-cultures, where we've co-cultured either peripheral blood or lymph node tumor B cells with previously healthy FRCs. And then we perform the RNA sequencing analysis following 3 days culture. So this differentially expressed gene analysis has revealed many deregulated genes, and I'll just highlight increased expression of alpha SMA, fibronectin, as well as various collagen family members. And this is indicative of fibroblast activation. And when we look at more closely at the gene pathways, we see an altered TNF cellular response, altered extracellular matrix remodeling pathways, interestingly, cell adhesion and deregulated angiogenesis and inflammatory responses, all being deregulated in these conditioned fibroblasts. So one important aspect of the laboratory is to actually assess the immunomodulatory consequence of these altered fibroblasts. Now, just very briefly, migratory dendritic cells that express CLEC2 are known to engage podoplanin on healthy FRCs. Now, this CLEC2 podoplanin engagement is required for dendritic cells to spread and become motile within the reticular lymph node network. And hopefully, this time-lapse video here illustrates a dendritic cell which is CLEC2 positive, spreading effectively and moving across the network. However, when we then use these modeling systems to look at the same dendritic cells on the podoplanin low CLFRCs, what we immediately observe is that the dendritic cells are not spreading and their movements are highly irregular or stochastic. And we will always quantify this in our assays. So just going back to patient tissue, you can see that the CD11C to mark the dendritic cells are showing a detached, I'll call it a derailed localization from the FRC network. And this contrasts quite dramatically with a non-malignant lymph node where the CD11C dendritic cells are in close interaction and are spreading effectively on the fibroblast network. And this cartoon just summarizes that what we believe is happening is that these altered fibroblasts are derailing dendritic cells, and this obviously has important consequences for immunity. So fibroblasts also have an important role in attracting, maintaining CD8 motility in lymph nodes. And this movie just illustrates that CD8s are moving effectively across fibroblasts. Just very briefly, the CD8s are inhibited in their speed across the CLL FRCs. And we've quantified this here. And this really led us to think, okay, what is the implication for immunotherapy, particularly the CAR T cells? So working with King's colleagues, we've been using an allogeneic CAR T product. And you can see that the CAR Ts are moving very rapidly on healthy fibroblasts. But when we used our modeling system here, the CAR T cells are quite dramatically inhibited in their movement across the protoplanin low CLL FRCs. And again, we're quantifying this here. And thinking about the mechanism here, what we're finding is that the altered fibroblasts in CLL show a downregulated expression of T cell chemokines. It also applies to dendritic cells, so CCL21, CXCL12, interestingly, and this contrasts quite sharply to the non-malignant FRC network in our control tissues. Okay, lastly, I'd just like to illustrate the concept of targeting fibroblasts in lymphoma and hopefully in CLL with immunotherapy, novel immunotherapy drugs, which hopefully can then activate the T cells, immune cells in this immunosuppressive microenvironment. So this video here, this is a 3D culture system. What we find is that the CD8 T cells are inhibited in their movement across lymphoma FRCs. This is actually high-grade lymphoma. However, when we treat the same model system with a novel fibroblast-activated protein, so FAP, IL-2 variant fusion drug, we actually are able to quantify that this drug is improving T cell activity and movement across the immunosuppressive fibroblasts. And this is our modeling data here. So really, this leads to the conclusion. So with this brief talk, I've highlighted that there are T cell defects, including impaired interferon signaling in patient T cells, which associates with a low infiltration of CD8s, and also associates with this, if you like, cold inflammatory state within CLL patient microenvironments. And we illustrate that inducing interferon signaling in the microenvironment, for example, the T cell compartment, this is the compartment we identified, can actually sensitize or reshape the microenvironment to being sensitive to these checkpoint inhibitor drugs. We've also been looking at remodeled fibroblasts in the lymph node, and we find that tumor cells are reprogramming these FRCs, and they appear to be acquiring altered immunomodulatory potential, which is inducing deregulated dendritic cell and CD8 T cell motility. So we believe this has important implications for both endogenous immunity and in the current immunotherapy era, particularly the CAR T cell data, which I illustrated. And ideally, what our aim is then to start thinking about targeting these immunosuppressive stroma cells in CLL and lymphoma to activate immunity and therapies for patients. So lastly, I'll just thank my team at King's, including clinical colleagues, peers. A big thank you to the Thessaloniki team with the collaboration, both Anna and Kostas. And in addition, I'll thank the funding sponsors as well as our industry collaborators, Celgene and Roche. So thank you very much.
Thanks, Alan, for this very clear talk. Are there any questions? We have time for questions, a couple of questions. I see anybody?
Martina.
Yeah. Martina?
Thanks.
So I have a question to the impact that you show that CLL cells have on the fibroblasts. So do you think that this is intrinsic to the malignancy of the cells, or what would happen if you take activated B cells or let's say a reactive lymph node? Would you also see that activated normal B cells impact on the fibroblasts in a similar fashion?
Yeah, very good question. So Natalia, who will briefly present today, so she's been working on this project. Okay, so we always do normal B cell controls, if you like, and we find that the activated B cells or normal B cells actually they do remodel fibroblasts, but they're upregulating the marker protoplanin. So that's almost like the opposite direction to what CLL is doing. So we believe this is related to the factors that tumor cells are expressing compared to B cells. Interestingly, all immune cells which are activated, T cells included, can remodel fibroblasts. So we're also exploring that, but obviously in a tumor setting scenario, the tumor cells was the first place to start.
Okay. Any more questions? Okay. Yeah. Okay. Thanks, Alan.
Thank you.
Thank you very much.